This video is on cell cycle control. When I say cell cycle control, I mean, how do we regulate cell division using internal and external cues? So how does the cell know when to divide? When cells are dividing out of control, that can lead to tumors and cancer. When cells are not dividing enough, that can be a problem as well. So we're going to start by talking about the internal signals. These are signals inside the cell. And the two main chemicals we're going to talk about are CDKs and cyclins. CDKs and cyclins are found inside your cell. CDKs are found at a constant level, and cyclins increase if you're doing things correctly in the cell cycle. Okay, and once cyclins increase to a certain point, they join with CDKs, and that pushes you to the next phase of the cell cycle. So basically, during the cell cycle, we have these checkpoints. And the three main checkpoints we're going to talk about today occur during synthesis, alignment of the chromosomes, and separation of the chromosomes. But basically, these checkpoints are points during the cell cycle which regulate whether or not you can move forward in the cell cycle. The default is off, meaning you can't move forward in the cell cycle. But as the cell prepares for division correctly, the cyclins build up. And this pushes this process on, and you can move forward in the cell cycle. Okay, so these CDKs and cyclins are really, really interesting. Okay, let's start first by talking about the molecules. And I'm going to show you some too. So we're going to talk about CDKs, and we're going to talk about cyclins as well inside the cell. As I said earlier, CDK levels stay constant and cyclin levels can rise and fall. So let's first go down here and talk about the molecules involved. Okay, obviously this isn't exactly what they look like. I'm just trying to make a point. Okay, so a CDK, that's short for cyclin-dependent kinase. So this is a kinase, and we know what kinases do. Remember, kinases phosphorylate other molecules. But this particular type of kinase can only be activated by cyclins, okay? So its default is off. So right now, this little kinase right here is inactive, okay? And there's a constant concentration inside your cell. So it's always present, but it's inactive without cyclins. Next, we have the cyclins, okay? Cyclins are regulatory proteins, and their concentration fluctuates depending on what's happening inside the cell. So basically, they cycle, right? And as the cell accomplishes its tasks, cyclin levels rise. And you can see, kind of reminiscent of enzyme specificity here, they match, okay? So you have these CDK cyclin complexes where a specific CDK can match up with a specific cyclin. Once they join together, they're known as a complex. Okay, so this whole thing right here is the CDK cyclin complex. Okay, and again, this is an example of an internal signal. Okay, so once this complex is activated by these two structures joining together, the cyclin and the CDK, now it can phosphorylate other molecules and other structures, pushing the cell into the next phase of the cell cycle. Okay, so we're going to look at this up here on this graph. So on this graph, okay, it's not a very good graph yet because we haven't labeled our axes. The x-axis here are our phases. Actually, let me use a different color. I don't want to use the same color as my cyclin. Here are phases of the cell cycle. And you can see them up here, G1S, G2, mitosis, G1 continues again. The y-axis here, we're going to label as concentration of CDKs and cyclins. Okay, so what's happening here is the cell is growing. We know in G1 we have growth. We know in synthesis we have DNA replication. We know in G2 we have repair. In mitosis, there's two things that are happening, right? So remember when I talked about the kind of checkpoints the cell is worried about. First, the cell is worrying about synthesis. Did the DNA replicate itself appropriately? Second, the cell is worrying about what's happening during mitosis. So if we have like prophase here, if this is metaphase, this is the phase where we're worried about alignment. And then if we have anaphase here, this is where we're worried about separation. So those are kind of the three main checkpoints. Cells have many checkpoints, but these are some of the big checkpoints that cells are worried about getting through correctly. Now, if you notice, and I'm going to use the same colors as before, our CDK levels here remain constant. But our cyclin levels can vary. So the question is, what do the CDK and cyclin complexes do? Okay, we'll start with the G1 complex. The G1 complex is a G1 cyclin and a G1 CDK that attach together. And once we have enough G1 complex, this helps phosphorylate the DNA. And helps phosphorylate our replication enzymes, activating them so we can move from G1 into S. So let me show you what this looks like. Okay, so here we have our cell. Here we have our nucleus. Here we have our chromatin. And what's happening is our CDK levels are constant. As the cell grows, we make a G1 cyclin. As the organelles replicate, we make a G1 cyclin. As the cell continues to grow, we make another G1 cyclin. As the organelles continue to replicate, we make another G1 cyclin. The CDK levels were always constant, but the cyclin levels, the G1 cyclin levels, increased. When you have enough of these, what happens is they phosphorylate the DNA, they phosphorylate replication enzymes, And that can push us from G1, where the cell is growing, into synthesis, where the DNA is replicated. So the chromatin is replicated then. At that point, the G1 cyclins break down, and you're left with the CDKs. Now, while we're on this, we'll go into the G2 complex. Now what happens is the DNA is replicating itself. So as the DNA is replicating itself, the G2 cyclin starts to be made. The DNA is continuing to replicate itself. The G2 cyclin is made. Then during G2, right, we proofread. We say, okay, has this DNA been replicated correctly? If it has, we basically reward ourselves and we're like, okay, let's make another. The cell rewards itself. Let's make another G2 cyclin. Keep proofreading the DNA, repair anything that needs to be repaired. If that's going well, we make another G2 cyclin. At this point, we have a new cyclin CDK complex. It's the G2 cyclin and the CDK complex. And what that does is that phosphorylates the nuclear envelope, helping it break down. Okay. At this point, too, our chromatin is being condensed into chromosomes. So chromosome. Okay, so we've made chromosomes. The nucleus is starting to break down. So that's pushing us from G2 into prophase, right? Ultimately into metaphase, but G2 into mitosis, okay? Because of this phosphorylation of the nuclear membrane, basically causing it to break down, okay? So at that point, again, the CDKs stay, and the cyclins, the G2 cyclins, break down. Let's write that down, and we'll go back to the cyclin CDK complex I really want to highlight, which is the M cyclin complex. So I said G1 complex. As we go through G1 correctly, this builds up, allowing us to move from G1 into S. So the G2 complex, as we proofread our DNA, as our DNA is replicated and we proofread our DNA correctly, the G2 complex builds up, okay? And this allows us to phosphorylate the nuclear envelope, causing it to break down. And this pushes us from G2 into mitosis. The last complex I want to talk about is the one I actually drew on this graph, because you would have lines for the G1 complex and the G2 complex on this graph, but I just want to focus on one specific example, and that's the M complex. And sometimes that's called MPF. Okay. MPF is basically increases as metaphase occurs correctly. So you can see right here, the CDK levels are constant. So we have constant CDK. But the M cyclin builds up as G2 prophase and metaphase are starting to occur correctly. Okay, so this builds up. And then when it peaks here, this pushes the cell from metaphase to anaphase. And then after the cell has been pushed from metaphase to anaphase, the cyclin degrades. And then the CDK is no longer active. So what's actually happening there? Basically, MPF is the name of the complex, the M complex. So MPF is a combination of the M cyclin and the CDK. Basically, as the spindle fibers attach correctly and chromosomes line up at the equatorial plate, in the middle of the cell, okay, we produce enough MPF, right? Because you're producing M cyclin as things are going correctly, okay? So MPF actually phosphorylates the enzyme that causes spindle fiber to shorten. and the chromosomes pull apart. So like I said before, when this happens, this is moving us from metaphase to anaphase. Okay. So if I showed you this on the cell, right? Oops. I gotta get rid of our nucleus. Our nucleus is gone. Okay. Again, we have this CDK. Okay. Here are our chromosomes. Here is our spindle fibers. As our spindle fibers are lining up correctly and helping move the chromosomes to the middle of the cell, that allows us to build up this M cyclin. The M cyclin joins with the CDK to form this M complex, right? And then once we have enough M complex, MPF, that phosphorylates enzymes, causing the spindle fibers to shorten, pushing us from metaphase to anaphase. So this only happens if we have enough, and then of course we have, telophase, we have our two nuclei. Then at this point, the M cyclin will break down. We'll still have CDK, right? Two nuclei, and then eventually two new cells. Right? So we're only moving from metaphase to anaphase if the steps leading up to metaphase are occurring correctly. That's what this M cyclin is ensuring. So that's a brief overview of the internal signals. You can have external signals as well. Okay. You can have nutrients and growth factors that are released by damaged cells to tell the surrounding cells to divide. You can have density dependent inhibition. This is when crowded cells will not divide. You can have anchorage dependent inhibition. Cells basically must be attached to other cells to divide. So go ahead. I've actually already written this down, so I'm going to put it up for you. Basically, external signals are when things outside the cell tell the cell to divide or not to divide. So like I said with nutrients or growth factors, let's say you get injured, damaged cells will release growth factors, or the cells will get nutrients if they need to divide anyway for growth. So this is basically for growth or repair. This tells the surrounding cells to divide to repair the gap or if you're growing. Examples are human growth hormone for growth or PDGF for repair. These are two signals. Okay. So this is telling the cell to divide. These two are telling the cell not to divide. So if the cells are crowded, they will not divide. Or if the cells aren't attached to anything, they won't divide. Okay. Cells do this by chemical messengers, right? When we talked about cell signaling, these could be different ligands that tell the cell not to divide. Cancer cells, however, do not follow these rules. That brings me to my last point. Okay. We have a lot of genes and proteins that regulate cell division, CDKs and cyclins, obviously, but there are other genes as well. One example is the P53 gene. The P53 gene produces a protein that repairs damaged DNA or leads to apoptosis, programmed cell death. So with a normal P53 gene, it's repairing the DNA. If the DNA can be repaired, it'll allow the cell to move from G2 into mitosis. If the cell can't be repaired, it'll program apoptosis, program cell death. So this is just one example. The P53 gene has been studied a lot because if you have a mutated P53 gene, you have a lot higher incidences of cancer like colorectal cancer and things like that. All right, I hope you enjoyed the video. Thank you so much for watching.